I'll press pause. Okay, so as a special treat, hello. Thank you. We've got 2 hours left. You'll be happy to know it's not entirely being used for lecturing purposes. Annette will be joining us at about 11 o'clock for the tutorial element of this part of the teaching. Okay, so we'll probably have a bit of immunology introduction from me for about a little over 50 minutes, then a little bit of a gap. As my mum used to say, you can go and powder your nose at that point if you wish to. And then there's actually a learning activity based on well-known card games, which is going to help you remember the functions of the various cells I'll be talking about over the next hour or so. Okay, so keep your concentration hats on. I'm going to compress effectively 2 lectures into one, but some of the reinforcement, in fact, will be with Annette and ourselves as you learn what a T helper cell does, how it relates to a B cell, and other things of that sort. Okay, so from today, think of multiple choice questions, think of short answer questions. So we're looking at the 2 main defense arms that are part of the immune system, and we'll also look at how those 2 arms collaborate together to defend us against disease from microorganisms, which most of you who've done higher biology will understand, but we'll also have a bit of a look at the process of when the immune system makes incorrect decisions and perhaps makes things worse, such as in the case of Ebola virus, where the really severe symptoms are in fact down to an immune overreaction. Okay, so that's what we're looking at today. How does it work? And occasionally, how does it not work? And the 2 main arms that you've probably heard of if you've done higher biology are the so-called innate immune arms. Their unifying feature is that you're born with them. They don't particularly improve over time, but in fact, I want to wave the flag for it, as it were, initially. It is in fact the heavy lifter as far as most of our microbial challenges are concerned. Then the second part is the so-called acquired part, which we'll come to later, which is classically known by antibodies being raised to a particular pathogen or cytotoxic T cells being raised to a particular pathogen. And the usual features there are the innate system has no capacity to deal with these pathogens. But if you think about all the microbes that are in our environment, there's no reason at all, in principle, why the bacterium that digests oak leaves in autumn as it lies on the ground, there's no reason in principle why it couldn't live inside the body, but in fact the innate immune system is very good at seeing them off. So if you stand back from the situation and say, who does the heavy lifting? It is in fact the innate immune system. Okay, so let's look at some very friendly to multiple choice question points in relation to this. So this is absolutely a fantastic feature. It protects against foreign cells or substances without having to recognize them specifically. Okay, so it doesn't have to spend a huge amount of time working out how to deal with it, as you're probably aware at the moment. In terms of coronavirus, that is clearly something that the innate immune system doesn't deal with very effectively, and it takes a couple of weeks for the acquired immune system to crank itself up to deal with that. Okay, so back to innate. It's there, you're born with it. It includes things like skin. It's inherited in that form. Doesn't require prior exposure, so it's more or less ready to go. And then the final part, which is both its strength and its weakness, is that it tends to recognize a general conserved property that marks an invader as foreign. And in that heading I would include that mystery bacterium that spends its time digesting leaves in the forest. It is actually in possession of surface molecules that, in evolutionary terms, our immune system, our innate immune system, has learned to recognize. And that's why very few of us go to the doctor with oak leaf disease, because in fact it's defeated by the system. Okay, so running through what innate immunity is all about, it's about our covering, it's about our secretions, and it is actually about cells. So it's an initial line of defense. Very few microorganisms can get inside our body except by particular strategies like food poisoning or whatever. So the intact body surface is fantastic by and large, and you'll be aware of course that if you injure the surface of the body, an infection is likely to result. Okay, so surface covering. Secretion of antimicrobial molecules, the stuff in tears, the stuff in sweat, which inhibits bacteria. Sadly, the stuff in sweat doesn't tend to inhibit the odour-causing bacteria, which may or may not be significant. Then we have mucus, up your nose, down your bronchi, various places which traps invaders. And in particular in your airway, that mucus, there's ciliary cells that actually beat and raise that mucus from the trachea, from the lung area, and into where it can be swallowed. And then the stomach acid is a pretty good destroyer of organisms. And kind of what I want you to hold on to at that point is 99% of whatever we meet in a day is dealt with by that kind of mechanism. And, you know, even if one of these fails, let's say stomach acid, for whatever reason, becomes inhibited, you do in fact open yourself up to some bacteria. Now that's fortunately fairly rare. Okay, so these are surface. Okay, so most of these have a phagocytic activity. Some of them are specialists in secreting inflammatory elements. You can imagine, I haven't just mentioned it specifically earlier, heparin's an anti-clotting factor. You could imagine that some invaders would find it handy to cause local clotting and prevent the arrival of acquired immune cells. So heparin is quite important there. And natural killer cells do not engulf, but they have this capacity to recognize that a cell has gone cancerous. And probably this is actually happening in all of us every day. Natural killer cells are jumping on cells that are beginning to look like cancerous cells. But they're also jumping on virus-infected cells. And I guess we would kind of say we're looking at PAMP type of situations there. Okay, so imagine you're going to play a game in about 40 minutes involving some of these cells, and you're going to find out what they're all about. Are there any questions about this so far? Would anyone like to argue with me on the basis of whether we can define something as a pathogen or as a non-pathogen? Are there any examples that you might like to challenge me with in terms of I said 95%, it might be 98%, might even be 99% of environmental microbes are of no worry to us because they're in possession of PAMPs. But occasionally they mutate and lose some of these qualities. So if you actually look at the microbial world, you can see bacteria which you only ever find in compost heaps, if you like. And you can understand that within a compost heap It's a very defined set of conditions. It's warm and damp and wet. But as you look at some environmental bacteria, as they, if you like, become less specialized towards these niche areas like the compost heap, actually do begin to look like bacteria that potentially can survive in us. And so there is a notion that pathogens can arise via mutation from environmental bacteria or viruses. I don't have an example. We have some, if you like, circumstantial evidence of bacteria of a particular class, some of which are pathogenic and associated with a recognized illness, and others which look comparatively similar, which are hardly ever seen as part of an infection. So that's an important idea to hold in your head. So we're about halfway through, I think. So I definitely want you to be able to talk about innate immunity and acquired immunity. So innate, the heavy lifter, your day-to-day friend, your HIV patient who has progressed towards AIDS, in fact, only survives because of the innate immune system. And you're probably aware that the kind of illnesses that the AIDS patient get are a very narrow range of cancers and particular infections that fully healthy people never have trouble with. And you can kind of look at the HIV patient as a model of what we need acquired immunity for. If they're kept away from pathogens, these days if they're given antivirals, they can live a perfectly normal life. But you note that they have susceptibility to cancer and susceptibility particularly to viruses and bacteria that most of us can deal with. And we deal with it on the basis of specific immunity. We require prior exposure to these foreign substances. In other words, we're not born with this capacity, but it plugs the gaps that innate immunity leaves. Antigen, well, that's easy to define. We don't necessarily say an antigen is something nasty associated with disease. As an immunologist, you'd say an antigen is any molecule that the host does not recognize as self. Okay, good working definition. Found in all classes of vertebrates except jawless fish. I have no idea why that is the case, but obviously jawless fish lack other features. And at one time was thought to be absent in invertebrates, but recent history suggests it may not be so. Okay, so it's there to plug the gaps. And because it doesn't know what the gaps are, if you like, it can't store the information for fighting smallpox or fighting coronavirus, unfortunately. It can't actually store that information. The antigens on the invader have to somehow prime the immune system to respond to them. Okay, so the invader, which by definition has got past innate immunity, requires to be sampled, if you like, and responded to. Okay, so that's where the lymphatic system comes in. Lymphocytes circulate in the blood. They're a type of white blood cell, but most of the time they're in the organs of the lymphatic system, things like the spleen, lymph nodes at various parts of the body. There's a network of lymphatic vessels which collects this lymph, which you may recall is kind of like the interstitial fluid in the body that's being drained through the lymphatic vessels. And you can imagine something that's separate from the blood circulatory system is, if you like, a drain for the tissues of every part of the body. If it's draining every part of the body, it's ideally placed to sample, to identify anything that's got into the body at any point. So it returns the lymph to the blood plasma, but in the lymph nodes, wherever they are, if it finds something nasty, it will start a response. And you'll be aware that in things like mumps, things like a whole range of infections, you get swollen lymph nodes. Okay, so something is happening there. Okay, so-called primary lymphoid organs. So we're tending not to think of all the distributed lymphoid organs. You know, if you get a nasty cut in your foot, you may find lymph nodes growing in your leg or wherever. But primary lymphoid organs tend to be things like spleen, thymus, bone marrow is a lymphoid organ as well. And that's where mature immune cells are made. And then these mature lymphocytes go to the secondary lymphoid organs, the various lymph nodes all over the body, and they're waiting there for potential invaders. So these lymphocytes circulate, go into the interstitial fluid, back to the lymphatic system, increasing the chance they will encounter their antigen. As you might imagine, trafficking in this area will be cells of the innate immune system. So there'll be neutrophils and other things cruising about. So you can imagine them jumping on all the non-pathogens, which will never get a chance to cause disease. Meanwhile, we've got our lymphocytes also in that kind of soup, sampling what's going on. So here's a general picture of these lymph nodes at various points. So if this lady got a severe infection, a mild infection, the lymph node might not show anything, but any deep wound, these lymph nodes would actually swell up, and we'd be having various lymphocytic cells proliferating in there. However, you can actually see what's happening in this one. Lymph enters a lymph node here. There are lymphocytes here, various cavities that will in fact fill with cells. If there's nothing going on, nothing invading, the lymph is returned to the general circulation. And here's another little list that you're going to conjure with later today. Hopefully this will. So we'll be looking at these later. Okay, what do you have to learn about these? T for thymus, T cells mature in the thymus gland. Cytotoxic T cells directly kill, they come alongside a target cell that's infected with coronavirus or flu virus or whatever, the factory producing all this virus, and they kill them directly. And they do that by recognizing the foreign antigen from the virus on the surface of the infected cell. And then, as I mentioned earlier, the helper T cell actually switches on the cytotoxic T cells and also activates B cells. Okay, so the T cell is really the heart of what's going on here. If somebody lacks T cells, their acquired immune system is utterly compromised. The antibody secreting cells aren't really able to get going. NK cells are similar to T cells in what they do in terms of recognition. So they're not working on the PAMP basis at the moment. They're working on antigenic recognition, but they also have the capacity to do these non-specific PAMP-based things. And what I'll just say about this point is we don't know all the types of NK cells are out there. They're actually quite a complex population. It would appear some of them can operate in an antigen-specific way and also in a PAMP kind of way. But then there are also specialist cells that seem to do one or the other. So watch this space over the next 10 years. Very important in pregnancy as well, the NK cell. Okay, so let's pause and reflect and go through what's just happened in terms of our about to be sick person. Why are you ever ill? it is usually because innate immunity has not been able to deal with the organism, and by definition that organism is a pathogen. Okay, everyone got that idea? So the acquired immune system is in there to deal with what is an evolutionary consequence of the guys stuck in the compost heap don't mutate to cause us a problem, but the guys with whom we share our skin or nasal passages, in the case of Staph aureus, may change enough such that the innate immune system cannot deal with them. So there has to be recognition of an antigen, and that's usually because it's still hanging around. We then need to activate the lymphocytes because, now here's a gross oversimplification, we are all of us in mid-childhood equipped with B cells and T cells that can recognize almost any possible antigen out there. So as a 5-year-old you will have had T cells for smallpox, B cells for yellow fever, sadly B cells for peanut butter in some individuals. And what we can say is that, and there's some gaps, it's probably between the age of about 5 and 14 we get the biggest range of B cells and T cells. And basically they're sitting there with a receptor for peanut butter or a receptor for yellow fever. Hopefully we edit out the peanut butter ones, but I'll come back to that in second year. And what we're relying on is we are, as a 15-year-old, in possession of B cells and T cells for any possible molecule out there, excluding our own, mostly. Okay, so you can imagine this. And scientists are a little bit uncertain about the numbers, but I would like to suggest to you that the naive person that has never met a particular antigen before might be in possession of 20 cells for yellow fever. It might be 200. It's actually very difficult in an entire human being to identify 200 cells. It might be 1,000. Anyway, what we do know is we don't actually have enough of them. So if you can imagine, if we had 100 million cells for yellow fever and 100 million cells for smallpox and 100 million cells for peanut butter or pollen or whatever, our lymph nodes would already, and lymphoid organs would be absolutely enormous. So we actually have, let's call it 200, just to pluck a figure out of there. 200 cells who are never going to be able to produce enough antibody. 200 cells combating an infection, no chance. 200 cytotoxic T cells able to defeat a cancer, no chance. They have to be activated, which means driven towards proliferation and then attack. Okay, so that typically takes 7, maybe 5 to 14 days. And at the end of 14 days, you begin to actually be attacking your antigen in some measurable way. Does that kind of make sense to everyone? Okay, so 5 to 14 days in terms of the coronavirus. 14 days is the isolation period. Probably in most people the peak inflammation is at less than 14 days. And we're having that inflammation and combating of the virus. And what we know about coronavirus is that we're relying on our T cells being multiplied up to actually directly kill virus-infected cells, plus a bit of antibody. And you begin to feel quite ill. And then hopefully by 14 days, you've won the battle. And as you're probably seen in the press, some people have virtually no symptoms of a coronavirus infection. Other people might have symptoms of mild flu, and then other people have symptoms of very severe flu. And the outcome, it would appear the mortality rate for coronavirus is something like 2 to 3% of people who are infected. Why doesn't it work? Well, again, we don't quite have it for some individuals. We don't quite have enough information, but we're not getting enough of this. And that's all I'll say. I mentioned that B cells have the capacity to recognize antigen. And if antigen binds to a B cell, in fact, very little happens. But if it's also bound to a helper T cell, that helper T cell will proliferate, kick that B cell, and as it happens, kick the cytosol you were born with. Okay, the detail of that, which again I've covered already. Activated B cells differentiate to plasma cells, and those antibodies do various things which ruin the day for bacteria and viruses. Cytotoxic T cells attack directly, and actually that's one important thing to think about. One of the problems with kidney transplant is that antibodies are not a big problem. So, why are antibodies not a big problem in kidney transplants? Well, that's a relative thing. But in actual fact, the reason that kidneys or hearts or whatever are rejected is not usually for antibody, which doesn't really get inside in quantity, but blooming cytotoxic T cells can actually invade your kidney in a transplant or whatever. Invade a cancer, which is rather more useful. And actually it's the cytotoxic T cells that cause organ loss. So the ability to dig into stuff is an important part of the defense of cytotoxic T cells, but it's really not helpful to the transplant surgeon. Okay, everyone kind of with me so far? So antibodies are lysing stuff, recruiting other cells. Cytotoxic T cells are killing virus factories. If that infection resolves, in other words, you're getting better, most of the plasma cells and cytotoxic T cells die by apoptosis. I mean, you've got hundreds of millions of them. You don't want to carry them around. If you're having new infections every day, you just don't have room to have hundreds of millions of the active cells. But there is a proportion that persist as memory cells, and like we've mentioned, if you think about it in a time scale, if instead of starting with 20 cells you've got 200 million or whatever, you're knocking days off the effective response time. Okay, so in detail, activated by antigens, and then I mentioned this, you absolutely need the helper T cell cytokines. They are a major defense against bacteria, viruses, and other microbes in the extracellular fluid, and, I suppose I hadn't mentioned this before, and against toxins. These are the nasty proteins secreted by some organisms. So actually neutralizing tetanus toxin, for instance, is an important preventer of illness. Yes, we'll look at that later. Learn this. Some of the structures here, just again, I would be familiar with the qualities of each of these antibody types. For instance, IgE is important in airway and gut. IgA is important in the gut. IgD, well, that's a fourth-year question. Again, we said there is summarising what the antibodies do. And I mentioned earlier, of course, like for a bacterium, the complement can actually lyse that directly as well. But that is to the side of what's going on here, and not specifically mentioned here, is the fact that antibody itself activates complement. Okay, another 5 minutes or so. Okay, quite a lot of intense information here. And we'll finish by talking about the T cell. Okay, so antibodies do lots of things. Antibody in free virus, very good at inactivating it. Free bacteria in your lymph or your blood. Antibody will do all sorts of useful things. Cell-mediated immunity. The top line I would like to suggest to you there, cell-mediated immunity deals with virus-infected cells. 100% of viruses that kill you will have replicated themselves in the patient's cells. The absolutely key step is to get rid of those virus factories. Okay, so remember that. T-cell receptors for antigens recognize typically a viral protein on the outside of an infected cell. They don't secrete anything like immunoglobulin, but they do have, and don't worry about this at the moment, they do have a similar receptor to B cells. And then what do I want you to know out of this particular slide? Okay, T-cell clone. T-cells also recognize the MHC class 1 molecule. Let's just leave that in your mind for the moment. And while I've talked about this business of killing off viral factories, the other thing I'd like you to bear in mind is the ability of T-cells to find their way through tissue. In some situations, antibody cannot get into, for instance, a virally infected zone. There's simply a circulatory problem. Sometimes coagulation is a problem. But T-cells, by and large, can make their way through, and that's an important characteristic. And as I mentioned 5 minutes ago, it's a very unfortunate characteristic if it's invading its way through a transplanted organ. The class 2 is found on antigen-presenting cells, and with this we're just going to finish in about 1 minute. You may wonder, how does a T helper cell get going? It needs to engage with an antigen-presenting cell that also has self-class 2 molecule. Now why is it there's a class 2 molecule? Well, it's a bit like the class 1, but it's also very, very specific to each one of us. And what this makes sure, and with this we're just going to finish at the moment, is that when a T helper cell sees the antigen from an antigen-presenting cell, it actually will engage with the class 2 and form this complex which switches on the immune response. So I'm going to finish with this. The T helper cell is the key to switching on B cells. It's the key to switching on cytotoxic T cells. If a T helper cell could be decoyed by free antigen, again, the immune system is knocked out. Class 2 is invented because when our T helper cell binds to antigen, it requires also to bind to class 2. Only circumstance under which that will happen is when it's against an antigen-presenting cell, like a dendritic cell or a macrophage. And it will only ever activate when it's alongside the cell it needs to activate with. Okay, I think that's the point at which I propose to stop. Some of the other features in here I'd expect you to know, and have a little bit of a look at this diagram. I would be familiar with that right up to about here. Okay.